Okay. Okay, welcome everybody. I want to thank you for joining us this morning. And so before we begin, I want to just a few housekeeping. I want to introduce myself, and I want to introduce Network Biotechnology. Especially if you are joining us for the first time, if you are seeing us for the first time, and then I will introduce Dr. Xia Shen, who is the main speaker for today's webinar. But yeah, so before we begin, I want to introduce myself. My name is Jeff. I am the general manager for Network Biotechnology. We are a mass spectrometry company that focuses on generating innovative data for metabolomics and proteomics. And we have been in this space for a while, and we have been developing our own database, our own technologies, in order to generate effective data from various matrices, from plants to animal models to clinical samples. And I want to showcase just a little bit about what we do. So we, again, focus on metabolomics, either from global profiling to targeted assays of various pathways, as well as proteomics, mass spectrometry-based proteomics, and as well as multi-omics analysis. And as multi-omics analysis, if you now generating data from genomics or transcriptomics, proteomics, metabolomics, it's on the easier end. But how to integrate those data together and analyze them in an innovative way is really the challenge in today. And also Dr. Shen will talk about how to leverage some of these data, especially the proteomics data, and looking at from a new paradigm, okay? So we do have a global presence. We have a main hub in our Boston facility, as well as R&D lab in China, in Hong Kong, Singapore, and in the Netherlands. So and if this is the first time that you are joining us at Metware, I do welcome you to reach out to us if you have any questions. And if you are considering applying metabolomics and proteomics, please come out and talk to us and reach out to us. So without further ado, I want to introduce Dr. Shen. Okay, Dr. Xia Shen is currently a professor at Fudan University at Greater Bay Area Institute of Precision Medicine. And Dr. Shen received his PhD from Uppsala University in 2012, and he had a short postdoctoral fellowship at the Swedish University of Agricultural Science. And shortly after that, he became a fellow at the University of Edinburgh, and he has held faculty positions in Sun Yat-sen University and Karolinska Institute. He's currently a professor at Fudan University, and his work has been focusing on developing statistical methods and approaches to quantitative genomics and proteogenomics. So it's my great pleasure to welcome Dr. Shen. And Dr. Shen, I give the floor to you.
Okay, thank you, Jeff, for the nice introduction. My slides are okay, right?
Yes, please go ahead.
So it's very glad to meet everyone here. It's my pleasure to be invited by Metware in Boston. I heard about their Boston lab being set up. It's very exciting. So we've been collaborating with Metware on some ideas, which is also my recent research, trying to extend the proteomic research to a new dimension or a new paradigm, what we call for the protein complexes. So my main research area is in statistical genetics or human genetics in general. So we started by learning the genetics of the proteins, right? So what we call proteogenomics. We map QTLs, we call pQTLs, and then trying to understand the shared genetics between diseases and proteins so that we can link the protein markers to complex diseases through genetics. So my idea today is first I will spend like 15 slides to introduce what we do or how do we deal with genetic data and proteomic data from our field of human geneticists. And then we go into the main new concept of protein complexes for today. So I guess all the audience today here will have heard about GWAS, right? So we essentially started by mapping the genes to the diseases and complex traits in humans. And sooner we realized we have to link the gene to the diseases through some molecular markers. So for today's topic, proteomics become the mediator for these genetic effects. And then through this, we try to infer drug targets, and we try to let these things benefit the general population. And then later on, trying to get larger data set and start doing this cycle of discoveries. So I will start by introducing some of our methods, right, that we developed in our group. So we are statisticians, so we spend a lot of effort trying to develop statistical models, analyzing genetic data, trying to understand the inheritance of complex diseases. And then I'll introduce our field of proteogenomics, trying to link proteins to our topic. We also have some contributions in the international consortium. And then later we go to the protein complexes as a functional executor and new biomarkers. So the overall aim is to uncover molecular basis or therapeutic targets anchored on genetics. So there's a lot of background. So I will try to simplify that by starting with something simple, right, in medicine. So I've been criticized, or our field has been criticized by some people, especially those bad lab biologists, so that people are questioning, why do we do GWAS, right, or genome-wide association study in large human population? We spend so much money, and what's the gain of that? One example is that we don't focus on individual discoveries, but if you look at the overall discoveries of the genes related to complex diseases, we realize having genetic evidence of the loci or the genes actually doubles our chance of a clinical success of the drug discovery as a drug targets. So it's some use of genetics, but unfortunately, this number hasn't changed much since 2015. Right? For the past 10 years, that's basically what it is. So genetics can only help us improve the chance of clinical drug discovery twice, but not so much more. So this is a great challenge in drug development, right? So of course, there's a lot of aspects we have to consider to make a successful drug development. But overall, it's a very high-risk project, about 10% if you start doing something like that. It's very high cost, average $2 to $3 billion per drug. It's very time-consuming. So in the recent Nature review paper, if you read about it, it says 25 years is the median time you need from a target to a successful drug. So it's a complicated procedure. It's very high risk, but as the vice president of AstraZeneca says here, right, a selective high quality molecule will never become a medicine if it's modulating the wrong target. So having the correct target will have a better inference of a potential target. It's actually very important for the procedure. So what do we do in genetics, right? So obviously genetics help in medicine or biology in general. So can we make more use of genetics, trying to do this better, right? Instead of just looking at the GWAS result, can we do actually more with the genetics that we learned? So it's a very hot topic, at least methodologically in our field, is trying to dissect genetic basis of complex traits or diseases through something we call summary association statistics. So basically GWAS results, right? Can we make more use of a GWAS result in a way that we can do better in biology? So to cut a long story short, I want to introduce our idea as this illustrative picture, right? So this is what's going on in my mind, right? If you want to develop a target or biomarker for a disease, of course, you have to have a cohort, right? So I think many of you will have a clinical study. You can have case control, compare the proteomics or metabolomics, and try to find the biomarkers. But genetically, we have a, well, not necessarily a better way, but a more fundamental way is to try to find the shared genetics between a biomarker and the disease, right? So we have one cohort where we have a protein GWAS, and another cohort we have a disease GWAS. The nice thing is that this can be two different populations, right? So from two different populations, we can learn the genetic architecture or the landscape of a certain protein marker. And from another population, we can learn the genetic basis of a disease. And when we compare them, if they share significant amount of those, we can have a sort of inference that this marker will be very important for the disease. And this is something what we call in quantitative genetics genetic correlation. So our group has been developing a lot of methods, we put a lot of effort at least in this field, trying to do this better, right? For instance, 5 years ago, we have this paper in Nature Genetics, trying to infer the genome-wide genetic correlation between traits. It could be a protein marker and the disease, only using the GWAS data, right? So you don't have to have access to the individual level original data. Just having the protein GWAS results and the complex traits results, you can actually infer the genetic correlations. And this year we have developed something locally, so that you can analyze genetic correlation in different regions in the genome, so that you can understand in more detail the shared genetic basis between a protein marker and the disease, or between two complex traits. And we also have a paper in press in, I hope it's soon going to appear in the journal Giga Science early next year. Something we call HDLC for co-localization. What it means that they try to identify the co-localization genetically or shared genetic basis between a protein QTL and a trait loci through genetic correlation, right? So it actually performs, according to our simulation in this complicated figure, better than existing methods. So it analyzes a lot of protein targets in UK biobank and some diseases. So these methods have been cited quite a lot in different journals, different fields, particularly in some cohort discoveries and epidemiology studies. And so this was the methods. We're not the only people doing methods in this field. You can have other ways to infer causality or genetic correlation between protein markers and diseases. But those methods are what we do. So for the analysis part, we have been organizing a consortium called SCALLOP. First started in Europe because it's called the Systematic Combined Analysis of Olink Proteins. And Olink was a Swedish company. So most of the European cohorts started using that. And now we expanded to many American cohorts too. We also have collaborators in the US. So basically, it's a large effort of human geneticists. We combine all the cohort data together, trying to meta-analyze the genetic basis of proteins measured from the Olink platform. The reason why we do that is, of course, at the beginning, Olink was quite expensive. And now it's not so cheap. So getting together more people will get more sample. And UK Biobank have 50,000 people with Olink proteins, so that now the sample size start increasing. So I've been leading two SCALLOP projects for understanding the genetics of the proteins. One is the genetics of ACE2 protein, the coronavirus receptor, during COVID. The other one was published last year in Nature Human Behavior for the neural-related proteins for the early Olink panels. So for those of you who are not familiar with what we do in human genetics, this is basically the simple procedure, or simple in a way, if you look at as a flow chart. So I, as the organizer for these projects, we design an analysis plan, and we distribute the plan to different cohorts that want to participate in the analysis. So they will execute my analysis protocol, then we collect all their results back as their summary statistics. Then we do some QC, we integrate all of them together, do some data curation. At the end, we have a final summary statistics from a big sample size. And then it's a more exciting part, right? It's the downstream analysis where we try to do causal inference, for instance, trying to find a potentially causal marker for protein marker for the diseases. So to give you some brief example, this is the one we did in during COVID, right? We did a big meta-analysis with ACE2 protein. There was 30,000 people at that time. We mapped the GWAS loci, and then we analyzed genetic correlation between ACE2 and other diseases and traits. So you can see from this heavy figure, but it's actually quite clear that for the cardiovascular diseases, it's mostly positively correlated with ACE2. Also for some psychiatric disorders, obesity, smoking, and even other complex traits. So you can see they highlighted on the top that the severe COVID-19 is highly or significantly, at least, genetically correlated with ACE2. So that the more ACE2 you have at birth or naturally, genetically, the higher chance you will get COVID severely as endpoint. And I try to do some causal inference between ACE2 and COVID at that time. So here I want to use this figure to explain the logic, right? What we do in genetics. So this figure shows the association locus, right? The area in the genome that's coding the ACE2 protein, so the ACE2 gene on X chromosome. And so the highlighted in red dots, those are the independent SNPs associated with ACE2. And because the y-axis is Z-score, so it has directions, we can actually do a meta-analysis of these SNPs. So you have this forest plot telling us the alleles that increase the level of ACE2 eventually will increase the risk of COVID infection, hospitalization, and also severity. So because these variants actually in the coding region of ACE2, right? So biologically, we believe they mostly work on the abundance or the levels of ACE2 in the human plasma, so that through the instrumental variable idea, we can infer there's a causality between ACE2 and COVID-19. In the last year paper, we did a lot of analysis of more than almost 200 proteins related to neurology. There are a lot of discoveries. I just try to show you two examples using the co-localization idea, right? These are coding regions of CADH6 and LGALS8. So this co-localization with a direction of genetic effects actually tells us the more coherent6 protein you have, the more chance to have mood swings as a complex trait. And the more galactin 8 you have, actually people who has more galactin 8 will have lower alcohol intake as their human behavior. So as review 2 of this paper summarized, right? This is an interesting paper that established a proof of concept. The circulating blood proteins may have utility as causal biomarkers for brain phenotypes. So you may question that, okay, the proteins may be in the blood only. How can it be related to brain phenotypes? But actually, it's actually quite nice. So genetics is a nice anchor, right? So even though the proteins, they function in different places or different tissues in the body, they actually most of the time actually share very similar at least common variant genetics in different cells. So because each of our cell has the same genome, so that the most fundamental genetic basis of the protein level is actually regulated by similar variants. So the shared genetics can actually be compared between brain phenotypes and proteins measured in the blood. So as drug discovery purpose, different people have different ways to do that. So in our paper, what we did is to take out our inferred targets, right? So the traits and the protein targets. And we're trying to match all of this to a drug bank database. So for the established drugs, if it has the same target as our protein and it treats the same disease as our disease or same side effect as our trait, we consider it as a matched pair. So you can see on the top left, there are 66 matched pairs in our analysis. And we realized for 80% of those, 52 of them, the established drug action direction, right, whether it's inhibitor or stimulator, actually matches our inferred genetic correlation direction, so that we can actually tell whether you should inhibit or stimulate the protein so that you can cure the disease potentially 80% correctly, which is quite impressive without even having some experiment, only look at genetics. So we're writing a review. It's not published yet, unfortunately. We took the job more than 1 year ago. Well, I guess you know how complicated it is to write a review for Nature papers. So it's going to be soon there, I think, in Nature Review Genetics. So if you are interested in reading more details of our summary of our field so far, this will be the place to go soon. It's called Proteogenomics in Human Populations. Okay, so that was the background, right? So I'm a statistician. We work on methods trying to understand or analyze genetic data to find shared biology between proteins and diseases so that we can infer markers or drug targets for diseases. So now I'm going to introduce some not new concept, but actually new way of dealing with the data that I think was the main interest of today. So we have different ways to measure proteins, right? So Medware, the company particularly, is very good at doing mass spec analysis. They have a lot of facilities both in China and now in Boston as well. I know they're also starting an Olink platform so they can do the antibody-based affinity proteomics. And another American company called, say, SomaLogic was recently, I think, bought by Illumina, was doing some affinity proteomics too, using aptamers. So all these technologies, they actually have assumption, right? So in our sample, say human plasma, we have individual proteins floating in the plasma, and then you trying to count, right, each individual one of them, different kinds of protein molecule, how much are they? At least you have relative abundance so they can compare. You can compare across samples. So this was the idea. But even though I'm not a biologist, I learned from some basic protein biology that there's a quaternary structure of proteins. So the protein, when they sort of got translated, it started with a polypeptide chain, and then you sort of get this alpha helix or beta sheet structures, right? That was the fragments you can actually target, not target, but untargeted measure using mass spec technologies. And then this targeted technology, the molecular tools can measure level 3 structure where they already fold into single protein molecules. That's what we have the names for instance, ACE, ACE2, those proteins. But at the end, those single proteins, or we call subunits, they actually form different complexes, and the complex can actually be very, very complex. You can have, a complex with more than 50 subunits, or even 100 subunits to be functional. So we believe, at least I believe, the next generation proteomics must go to the analysis of protein complexes, because those are the real proteins, so to say, in our life. So to give you some examples here, right? So I believe protein complexes are the real executors of biological functions. So on the right-hand side, you see myosin, hemoglobin, right? Protease reaction, antibody mediated immune response, all those things that we normally heard about. They are actually, because these things are also called proteins in Chinese, in Mandarin language, but they are actually protein complex, right? There are different protein subunits formed together so that you can become a functional unit. So this is an illustration. I'm not saying that it's a real observation, but you can imagine, right? So in our body or body fluid or blood, at least, there's all sorts of different shapes or different forms of proteins that are being there. And quite some of them are protein complexes. Because, for instance, the first one you see in the slides, we have all sorts of different vesicles, right? I think there are people studying exosomes, for instance. or extracellular vesicles in general. There are quite a few of those. It could be millions of them in one, a tiny bit of the blood. And we have also other type of complexes. It could be a substructure of a big complex, could be different sort of things. So the proteins actually interact with each other. Where in general, the protein-protein interaction is the eventual thing we try to measure, we should measure in the blood. So why do we need to measure this, right? Why only measuring the single protein cannot be enough? So I made a small video to show you that. So say we have a protein X, it can form different kinds of complexes, right? One is called XA, one is called XB, and XA is functional in the brain, XB is functional somewhere in our body. Then different people could have the same level or similar level of protein X, right? So we cannot actually see a difference between people, but it could be the levels of the complex XA and XB, right? The relative proportion of the complexes that they form that can change our diet, change our behavior, and eventually lead to diseases. So if this happens by only looking at protein X, we of course wouldn't see it. And if you only analyze protein A and B, maybe it's other type of complex involved, so it's not so clear after all. So we have to deal with the complexes, or at least analyze those things. So to give you one example, this is something I learned recently. I know two professors in China. They are quite into protein biology. They are, of course, better biologists than me. Both of them are studying a protein called PI3K. Obviously, for people who know this protein, it's a very important disease marker. But for instance, we know that PI3K, they can form different complexes. If we form a complex with AKT, it becomes a very important marker for cancers, right? The increased abundance of the complex will be associated with various cancers. And if it forms a complex with IRS1, it becomes something related to metabolism, right? So the loss of function of this will be associated with type 2 diabetes, for instance. So if only look at the protein in general, we wouldn't see this. You have to looking at the complexes. So I would argue that protein complex is the eventual executor of life, dynamic indicator of health, and effective targets of diseases. So obviously, as I introduced, the current platform, they all have a limitation, right? So they focus on protein subunits, even though the proteins may already be a complex form, right, in the sample, but the targets are designed to measure subunits or individual proteins rather than the true complexes, regardless of technology platform. So here I would like to share with you my motivation for this idea, or something I'm going to introduce to you, is can we quantify protein complexes, right, or at least score them, phenotype them, using the established proteome assay? Because developing, I know there are people developing ways to target simple protein-protein interactions, right? Whether they are there or not, but to quantify them across large sample and to have a multiplex different combination of protein complexes, it could be very complicated. So can we do something with the current data, right? So we have a very solid proteomics data so far, especially using some established platform. It's quite good. So can we make more use of that to infer protein complexes? That was the motivation. So to do that, this becomes so-called the deconvolution problem, right? So we have to solve an equation from the proteome data to a protein complex score or some new phenotype data. So to do the deconvolution, of course, we need the information to link protein complexes to proteins. So there are not only two, we actually have a couple of different sets of information we use. Of course, the biological pathways, right, the established knowledge are important. But just to share with you two very important information we use in our model. One is AlphaFold 3 predicted subunit connectivity. So this is free. At the beginning, it was not so easy to use. It's quite limited, but now everyone can use API to access AlphaFold 3. So when you give two sequences of proteins to AlphaFold 3, trying to predict the complex or the combined structure, they sort of give you a structure and they give you some sort of scores, like here it's IPTM score or PTM score, to tell you how confident this structure is, right? So this tells you the structure somehow, but doesn't tell you the abundance, of course. So what we do in our model is not to use the structure itself, but try to use the confidence it gives for the structure, right? So when we do a lot of that, we will start having all sorts of different combinations of proteins, and AlphaFold 3 will give us some structure and some confidence. So some structure may be very, very low confidence, is actually not there, but its score is important, right? When we build up the matrix of all these confidence scores, we use this as prior information in our statistical model, and then we try to learn things from that to infer the existence of protein complexes. So this is one thing. The second thing we did is that we actually bought some protein subunits in the lab, and we synthesized those proteins into protein complexes by knowing the database, right? The database tells some structures are there. And then we try to spike each of the protein complex into a standard plasma sample, and we measure the proteomics, say using oligosomal logic from that, so that we learned how the protein complex will affect the readouts of the proteome. So we have a fingerprint of disturbance of protein complex on the proteome data. And then from this fingerprint matrix, what we call, and the proteome data and the AlphaFold thing we did to tune the model, we build up a model through an algorithm so we can learn some new data or infer some new data from the proteome information, and we call it Complexome data. So we have different scores for different complexes and for different samples as well. So to show you what we did, we didn't do that many complexes, but we actually did quite a lot of that. We also have some imputation and other things going on in this math just to fit in this matrix so that we can use the whole information in the downstream model. So to cut the long story short, right, all these hundreds of millions of data points, when we have that, then we build a statistical model, or if you know some statistics, we have a likelihood-based model, so we try to maximize the likelihood of observing our protein data by giving some protein complex scores, right? So we try to use an algorithm to guess what kind of protein complex score you should have in this particular sample in order to give you the protein data you measured. So from statistical model, also some what you call machine learning or deep learning algorithm, we build a big thing. It's like ChatGPT, right? It generates text, but here we generate protein complex scores. We have a white paper. I'm going to show the website later describing the mathematical logic behind that. But it's basically a big model to try to turn proteome data into protein complex data. And we call it confident complex fingerprint deconvolution technology. So now I've introduced the idea, right? So why we need to analyze protein complexes, what was the motivation, and what exactly we did statistically or computationally as a tool to make more use of proteome data. But does it work, right? So the immediate question is that, does it work? What I have to say is that the outcome, what we output as protein complex data, cannot be totally perfect, right? Because some of the protein assay even doesn't work perfectly. And some of the complex information, of course, with low confidence, some is higher confidence. But we have a couple of examples. Actually, we have quite a few examples. We're writing papers about a lot of these. I'm going to try to show you some examples. And hopefully after this, you'll be convinced that most of the analysis and at least some of the markers really works as complexes, which give you something that proteomics traditionally cannot tell you. So the first example I'm going to show you is from a real clinical case. So this is a hospital in China. So it's a skin department. It's called the dermatology department. So they have a lot of different patients. Some of them have, skin immune problems. Some maybe have a rash. Some maybe itching. And they also have some even more severe cases like skin cancer. But in this case, it's for the diagnosis of skin immune symptoms. So we have more than 100 cases, 60 controls in this sample. So all of these went through Olink inflammation 96 panel. So that you get 96 inflammation markers or proteins from the Olink platform. These are very good measurements because we do have in our Scallop consortium very solid genetics papers based on this data, this panel in our cohorts. So for this case, what they want to do is that does this inflammation markers tell us who have potential skin immune problems, who does not, right? Because not most people have symptoms. Some may be on the way of having symptoms. Some may have mild symptoms. They just want to cluster, right, patients into the correct group, whether it's a disease or not. So after you do the proteomics, you have this 200 or 176 people by 96 protein matrix, and you do a principal component analysis, right, unsupervised, try to group them. And on the right, you can see it's actually not bad, right? The only proteins are not bad. They give you some sort of ability to tell the two groups. It's 85% accuracy according to AUC. But you can see that by telling some of the controls are mixed with cases, so that you not always can tell most of the cases from the controls. So what we did is that, so this is a completely third-party validation, right? So what they do is that they have their data, they send to us, we run our algorithm, the confident algorithm, trying to generate 203 complexes through the 96 proteins. So we get a new data set with 200 complexes. We send the data back, right? And they did the new analysis trying to group the patient. So you can see, obviously, without even looking at the original 96 proteins, only the 200 complexes can already tell the two group of patients very, very well. So we almost perfectly, 99.8% accuracy, tell the two group which are cases, which are controls. So this shows that at least through this panel, right, for the skin problem, we can do clinical diagnosis. We can subtyping potentially different patients, and potentially some of the markers could be used for later analysis of, say, drug response, treatment response, and some other uses. So as a demonstration here, you can see that this technology can be used to extend the proteome data in general, right, to give more utility of prediction. And then as human geneticists, right, as I introduced, what we do is that we try to understand the genetics of proteins, and then we try to find biomarkers. So now we have the protein complexes technology. Of course, we can analyze the genetics of the protein complexes instead of the protein themselves. So we did that in our cohorts in Edinburgh. To start with, we choose a cohort called ORCIDs. It's called Orkney Complex Disease Study. It's 1,000 people sampled from the Orkney Islands in the very north of Scotland. The reason why we choose this cohort first is that we were so familiar with this cohort. Secondly, that these 1,000 people has very deep phenotyping. They have all sorts of different omics measured, genomics, of course, measured, Olink data measured, so we can directly use our algorithm. So the more than 1,000 Olink proteins were transformed through our algorithm into around 2,000 complexes. So now it's all about analyzing 2,000 new phenotypes and see what we get. We get many different things. I don't have time to introduce to you all of those, but to give you some examples, for instance, this biomarker, right? The PLOR complex and the CDH5 FLT4 KDR complex through our analysis are very significantly associated with diabetes cases and insulin intake. But none of these subunits, the individual proteins, are associated with that. So we haven't validated this in our other cohorts yet, but potentially this gives you complex biomarker predict onset of diabetes more than 10 years in advance, because these samples were sampled more than actually about 20 years ago. And the plasma in the freezer, right? So we analyzed the new data from that. And to give you another example, this is even more impressive. On the left, if you look at the two protein markers, right? What's called MASDC2, what is called NUDT5. These are not associated with the trace on the x-axis, which is the father's age of a heart attack. This is, of course, very challenging, right? So we're trying to use the children's protein marker measured in the plasma to predict his or her father age of heart attack onset. That's already very challenging, but it's still doable because they're relatives. You can see through the complex analysis, we have a new score for the two protein complex, and it's very significantly correlated with father's age of heart attack. Particularly, if you look at the box plot, it's a very good marker to tell to predict the very early, a young age of heart attack, like 30-ish people's risk. So overall, we have a lot of discoveries. Many of them are shared between proteins and complexes as a phenotype level biomarker. Some protein markers, of course, missed by complexes because they are not the same. But most importantly, we do have 48 complex markers like this that are only detectable through the complex analysis, not discoverable through the protein analysis. And we did some genetic analysis, to 2 slides about that. So this one shows 2 protein markers, right? If you don't look at the complex in the middle, right, we will see the increase of CD177 genetically will decrease the risk of lung cancer. So if you follow up this as a drug target in pharmaceutical companies, if you think, okay, if we increase the level of CD177, we will have potentially be able to lower the risk of lung cancer or treat lung cancer. But actually, through our analysis, CD177 can form a complex called the CD177 proker, and the cis region, the local genetic correlation pattern, you can see more similar to lung cancer. So if you inhibit, if this is true, right, the complex is actually the biomarker. We should not stimulate CD177, but rather we should inhibit this particular CD177 complex to cure lung cancer. On the right-hand side, you have ulcerative colitis. Is a similar situation. So without looking at the complexes, you may actually have we have the right target, but have the wrong inference of the direction, so that the follow-up analysis will be wrong. This figure looks more complicated, but it's actually quite simple and exciting. So on the left, you have 2 QTL for the CA13 gene. One is the eQTL of the CA13 in the blood, and the green one is CA13 protein pQTL in UK biobank. So here you actually see the 2 associations, right? The genetic variant controlling the CA13 gene expression and protein expression are actually not the same variant in the coding region of CA13. So most of the time, we don't know why this happens. Of course, through the central dogma, this is weird, but it could be different things going on. It could be protein complexes. It could be, glycosylation. It could be different isoforms. All sorts of different things. But here we guess we know why. So CA13 protein forms 2 different complexes as we predicted. One is called CA13 compt, the pink one on the right. CA13 ANASA4, the green one on the right. So according to this, you see a CA13 compt complex co-localized, right, quite well with CA13 gene expression, which means most of the CA13 gene, the transcript becomes the compt complex. But the O-link antibody here actually only targets the CA13 ANASA4 complex, so that when we measure CA13 protein using O-link assay, we only see the green association that's colocalizing with the green signal. So what this tells us is that we are targeting CA13 when we measure protein, but actually because of the bias, so to say, in this particular case, we are actually targeting the CA13 under the 4 complex. And this is so important because for the downstream analysis, when we design a drug, we should not be looking at the CA13 structure, but rather the CA13 under the 4 structure. And they give you completely different targets, so that completely different chance of being successful in drug development. So to summarize, if you only look at the complex marks we found, right? So more than half of them are consistent with drug action if you match with the drug database. Among them, only 13% of them are consistent through the protein marker only analysis. And nearly half of them cannot be discovered through traditional proteomics. So this shows the importance of analyzing protein complexes, at least using our current algorithm. So this was all the scientific part. So at the end, I will try to introduce our company that's collaborating with Mathware in Boston so far. We have a headquarters in Stockholm, established last year, called Quantics Biosciences, a spin-off from Karolinska Institutet. We now mostly work in Guangzhou, trying to develop some data analysis algorithm. I have a branch here. So our product is, of course, in the narrow sense, right? Our product is to extend the state-of-the-art proteomics. So if you have proteome data from different platforms, of course, our training was done in Olink SomaLogic mostly, but the AI prediction through AlphaFold can actually be extended to, of course, MassSpec as well. So if you have different platforms, you can extend your proteome data into protein complex data. For instance, if you have SomaLogic data, we can extend to more than 3,000 to 4,000 complexes or phenotypes to your data set. And for Olink small panels, we can extend 1 to 300 complexes for the target panels. For the Explorer panel, of course, we can do 3,000 to 4,000 as well. So if you're interested in this, you're happy to contact either us or Mathware in Boston. So you can get this tested in your data set. If you visit our website, there's a try now page. If you click on analyze your data, you will see a portal. In this console or portal, you can upload your own data. Of course, if you have ethical permit, we will be processing your data, give you some reports, and so we have an online platform to handle your project if you are interested in. So that's all I have for today. I think for people in the US, it's better to visit our website hosted in Sweden, quantics.se. Of course, the Chinese website metaproteome.cn is also working. If you can access that, the console's there. And if you want to get in touch with us, you can either contact Mathware or us through email, info@quantics.se or contact.quantics.limited China. Okay, thank you very much.
Well, thank you, Dr. Shen. Thank you so much. And I'll leave this slide up just a little bit more. And I'm not sure if the chat box is working via the YouTube live stream, but there are some questions that maybe I can ask, and then we can have a little bit of discussion. So as you have developed the confident algorithm, and a lot of the example that you have shown are protein complex with 2 subunits, and I assume that it does extend into 3 subunits and 4 subunits, and it becomes a more of a combinatorial problem eventually. How would you please comment on how well your algorithm can scale to even larger complexes?
Yes, that's a very good question. Thank you for asking. So as you say, right, it's a combinatorial problem. So that if we consider all the combinations, right, eventually we will, it's becoming too much, right? So we can't deal with that many so far. One thing is that So you see we have two sets of training data, right? One is on the experiment side, we try to synthesize some complexes. The other part is that using AlphaFold to predict the structure. So for the wet lab experiment part, we have a database for the established complexes, right? So already known from literature. So what we did is that we tried to focus on the protein markers already covered by the current Proteome platform, right? Like Olink, SomaLogic, those panels, and trying to see which of the combination already can assemble some sort of subunits or subpart of the known complexes. So that's why we choose those things. Of course, we choose 2 or 3 because it's simple. If we don't want to make it too much. And for the AlphaFold prediction, of course, like you say, if you predict all the pairwise combinations, it's handleable so far for thousands of proteins. For triplets, we can handle some of them if those are known complexes. Of course, we're trying to do that. It's better. But if we do all the triplets, it's already too much. If we have all the quadruple combination, you can imagine it become too much. But eventually, I think we can get there. Unless we find even smarter ways to deal with the algorithm, we will try to extend to more subunits. But so far, I think for 2 subunits, right, the double combination and triplets combination, we have a good coverage. Yeah.
Well, excellent. Thank you so much for that. And so far, I mean, your focus has also been with drug discovery. A lot of the data are from clinical samples and clinical data, and a lot of researchers are doing basic research in mouse models, rat model, or other model organisms. And is the algorithm suitable for other model organisms as well?
Yeah, of course. It's so this is just a tool, right? It's like a mathematical machine, right? So what all it does is to generate new data from your sample. So there's nothing biased towards sample type. So as long as your proteomic platform can spit out proteome data from the mice, from the even plants, right, or humans, you can of course predict the complexes through that. Of course, how good the data is for a particular problem, you have to try that. But I hope through today's examples, it's people can be convinced a little bit that it's going to work sometimes.
Yeah. Does the algorithm needs to be retrained for every species, or what do you feel in terms of expanding it to model organism, or does the model work relatively well for some of the well-known model organisms such as mouse?
Yeah, that's a good question too. So we actually when I showed the extension of the current data, right, you see we have 2 sets of complexes. One is AI predicted, one is synthesized. Actually, most of the working or after QC, passing QC complexes are the one predicted from AI. So so then I think regardless of platform, I think it can be generalized to all sorts of different species, right? As long as it can be predicted. So I think the AI, the AlphaFold part of our algorithm will work for different species. We will try to improve. I know if you apply, say, human Olink platform to mouse or even monkeys, it's going to work for some proteins too. So they all depends on how good the protein measures are. So as long as the proteins are measured with good quality, it's okay.
Well, excellent. Well, I want to appreciate. Again, I don't know if the chat box is working via YouTube, and we will have the video recorded and that you are able to play back at your own time. So it will become a webinar on demand. I do appreciate everybody joining us this morning, and I want to thank Dr. Shen again for his time and presenting us this new paradigm of not only looking at individual proteins or individual protein subunits, but looking at it from a complex perspective. So I do appreciate your time. And again, if you're welcome, if you have any questions, feel free to please reach out to both Network Biotechnology as well as to Quantix. And if you're wanting to have existing data from proteomics, either Olink, SomaLogic, and you want to apply some of these analyses, please reach out to us. So this will conclude today's webinar. And I do again thank you for your time, and have a great rest of your day. Thank you, Dr. Shen.
Yeah, thank you, everyone. Thank you. Cheers.